The overall goal of this sample preparation method for MALDI MS is to reduce the spatial heterogeneity in ion signals during MALDI MS.
A well-controlled drying environment was built to optimize the conventional dry droplet method by manipulating the hydrodynamic flows inside the sample droplet during the drying process. The main advantage of this technique is that the spatial heterogeneity of the ion signals for the sample prepared by the optimized dry droplet method can be effectively reduced. Though this method can provide homogeneous samples for the THAP metric system, it can also be applied to other metric systems, such as alpha-cyano-4-hydroxycinnamic acid.
Generally, individuals new to this method will struggle because controlling the drying condition is not easy.
To begin this procedure, open the door of a drying chamber and immediately place a previously cleaned sample plate on the copper base block. Then close the door. Manually adjust the gas flow meter to set the nitrogen flow rate to 10 standard cubic feet per hour. Following this, fine-tune the gas flow meter to ensure the relative humidity is always below 25% in the drying chamber, as measured by a hygrometer. Using K-type thermocouples to monitor the temperature, adjust the water circulator temperature manually until the sample plate reaches 5 degrees Celsius for the experiment. Ensure the required temperatures and the relative humidity are reached before